Hello, and welcome to the second part of our lecture for inflammation and repair. This time around, we'll be talking about the second phase of acute inflammation, specifically the cellular events or the events wherein the WBCs are participating in the acute phase of inflammation. Now, first, we'll know what happens, how the leukocytes move from the blood to the tissues, and um, we'll be introduced to special molecules known as selectins and integrins. Okay, we'll also know how um, the WBCs move from the peripheral tissues to the actual site of injury in a process known as chemotaxis. And once the WBCs encounter the inflammatory agents, how they gobble them up in a process known as phagocytosis. Now, after they have ingested the microorganisms, we'll also look at the mechanisms by which they kill um, these organisms, and we'll also be exposed to a few pathologic entities or a few diseases wherein you will have defects in WBC function. Now, as a review, we know that acute inflammation is composed of two major events, the vascular phase, and that is composed of a transient vasoconstriction process, and then vasodilation, and then after, there is leakage of fluid out of the blood vessels. Now, after the blood vessels play a role, mag-shift ang starring role to the WBCs. Now, the function of the cellular phase of acute inflammation is to essentially bring the soldiers of the body, the WBCs, to um, the area where they are needed most. And this is brought about by a process known as leukocyte extravasation. Okay? And once the WBCs are delivered to the site of injury or the site of inflammation, um, there is activation of the WBCs wherein they actually perform their functions in host events. And what are these functions? First, they eat the offending agents, they kill the bacteria and microbes, and they clean things up. After nilampat yun ang organisms, they will get rid of the dead microorganisms and the dead cells na collateral damage sa infection. They may also induce tissue damage and prolonged inflammation, since the leukocyte products that destroy microbes can also injure normal host tissue. So the point here is that inflammation is non-selective. Okay, so it will damage both the microorganisms and normal tissue as well. So there needs to be a mechanism in place na makontrol yung mga inflammatory process when it is no longer needed by the body. Now, as a very quick overview lang, how does the WBC remove the offending agents? First, there needs to be a mechanism na ma-recognize ang microbes by the WBC, and it does so by receptors present sa surface sa WBC. Now, once ma-attach siya dito, the leukocyte is said to be activated. Okay? Ma-activate ang leukocyte, and this facilitates the activation of enzymes. How does it activate enzymes? These enzymes, particularly protein kinase C and phospholipase A2, this is brought about by changes in the plasma membrane permeability, causing an influx in calcium. Now, we know na one of the reasons why the cells die when we talked about cell injury and cellular death is that an influx of calcium can facilitate activation of enzymes, and this is what happens here. So, ang metabo, when an organism encounters, when a WBC encounters an organism, and there is an interaction between the microbial cell wall and receptors present sa surface of the WBC. This will lead to conformational changes in the plasma membrane, facilitating the entry of calcium. Okay? Once ma-enter ang calcium, ma-activate ang enzymes, particularly two very important enzymes involved in the signaling process of the cell, protein kinase C and phospholipase A2. With these two enzymes activated and their associated cell signaling mechanisms are activated, there is destruction of the organism, first by eating the organism and killing the organism while it is inside the cell. Okay? Now, this diagram essentially shows us a very detailed description of what happens during the recognition phase of the cellular events in acute inflammation. So, the first question is, how does um, a WBC um, recognize recognize microorganisms? It does so by certain groups of molecules found in the cell surface of microorganisms called PAMPs, or pathogen-associated molecular proteins. Now, PAMPs are non-specific. It can be any molecule, but usually these are molecules that are composed of either carbohydrates or lipids. Okay? And this is particularly true for bacteria, which have an outer membrane composed of lipopolysaccharides. So in most bacteria, particularly in gram-negative bacteria, they have a layer sa cell wall nila called lipopolysaccharides. Now, these lipopolysaccharides will act as PAMPs, or pathogen-associated molecular proteins, which will interact with receptors in the cell surface of the WBCs. And these cell receptors that recognize PAMPs are known as TLRs, or toll-like receptors, TOLL, toll-like receptors. Now, I'm going to skip the nitty-gritty of the various signals, but the end result is that this will activate the production of certain cytokines, particularly TNF, which in turn leads to the production of interleukin 1 and interleukin 8. But we will focus on interleukin 1 as the most potent of the three. Okay? Now, interleukin 1 is produced by a protein complex. So essentially the whole thing here, with the end result being the production of interleukin 1, the whole thing is known as the inflammasome. Okay? The whole thing is the inflammasome, and ang purpose of the inflammasome is to secrete interleukin 1, which will kickstart the entire process. Okay? Now, what's going to happen is, how does the WBC get from inside the blood vessel to the specific site of injury? And basically, you have these steps. Okay? So first, of course, where did we stop last time? Diba, ang nahitabo is, ni-dilate ang blood vessel, ni-hinay ang blood flow, ni-gawas ang tubig. Right? Now, think of a hose where you have pebbles inside the hose. You turn on the faucet, and these pebbles, by virtue that they are floating within the water within the hose, naase sa tunga. It will go through the hose. Usually naase sa tunga yung mga small na particles, and it will flow through the hose lang. Now, while the water is running, if you make small holes sa hose, what's going to happen is water is going to leak out. And because water is rushing out of the hose through these holes, the small pebbles will slow down. Tinigawas man ang tubig. So nag-slow down siya, and instead of being in the center of the flow of water, since nihinay man ang water, kinigawas man siya, it tends to slow down and approach the walls of the hose. Essentially, that's the same thing happening dire. Okay? Since nigawas man ang plasma sa imuhang blood vessels, what's going to happen is the cells that should have been found at the middle of the blood vessel will slowly marginate. Okay? It will slowly marginate, and it will start rolling across the blood vessels. Mutambal siya across the blood vessels until muhinay siya, and then stop Mupilit siya sa blood vessel. This is known as adhesion. Now, once mupilit na siya, mulusot siya sa endothelium, and this is a process known as diapedesis. Okay? So, nakagawas na siya sa endothelium, naana siya sa tissue. How does it go to the specific area? It goes to the specific area in a process known as chemotaxis. Okay? So here, you slow down, mag-marginate, mag-roll, mupilit, mo-release, mupilit, mo-release, mupilit, muhinay-hinay siya until ma-stop siya, mupilit siya. This is known as adhesion. Mugawas siya. This is known as transmigration or diapedesis. And then it will move to the specific area in a process known as chemotaxis. Okay? So this photo shows us what happens in margination. So here you have neutrophils lining the edges of the blood vessels, and this again is brought about by the slowing down of blood. Normally, when you have when you have floating cells within plasma, as long as blood is freely flowing, naagi niya siya somewhere towards the center. Okay? If the blood is freely flowing. So the lighter ones, like the red blood cells, are in the center of the flow, and the heavier ones, like WBCs, are slightly off-center. And this process wherein you have different columns of fluid passing through a tube is known as laminar flow. Okay? So this is the process. Again, another picture of the process. Now, what are the molecules that are responsible for that? Now, you have, for the rolling process, you have molecules known as selectins. Okay? Known as selectins. And these are produced by the Weibel-Palade bodies that are found within the leukocytes. Okay? Selectins are found on the surface of the blood vessels. Okay? So, selectins can be found in the surface of the blood vessels and in the endothelium. Okay? So, sa WBCs, you have two kinds of selectins. You have in the WBCs, you have selectins known as L-selectins, and in the endothelium, you have two types of selectins, E-selectins and P-selectins. Okay? So mag-interact sila. Think of selectins as scotch tape. Mupilit siya pero dili kaayo siya strong. Okay? The selectins will bind to their ligands. Okay? The selectins will bind to their ligands, and the ligands, or ang molecules kung asa mo attach ang selectins, are known as sialylated Lewis X glycoproteins. Okay? Sialylated Lewis X. So selectins plus sialylated Lewis X, this will facilitate rolling. So mura siyang scotch tape, pilit siya pero dili kaayo adhesive. So ang hitabo, it will only serve to slow the WBC down. Okay? And because of that, mo roll siya. Slowly, it will tumble sa endothelium. Okay? Now, once it has slowed down, what's going to happen is that the WBCs will secrete another receptor known as integrins. Ang integrins serve to facilitate definite attachment. Okay? It will serve as the definite attachment. And ultimately, ang mahitabo, muhunong ang WBC from rolling. Okay? So these are known as integrins. Okay? So again, muhinay ang blood flow, there is margination, muduol siya sa blood vessel, there is rolling through the action of selectins and sialylated Lewis X, and then mustop siya. This is a process known as pavementing. Pupilit siya sa blood vessel wall, and this is facilitated by integrins. Okay? So, nihunong at this point, nihunong na siya. Nihunong na ang pag-move sa WBCs. Okay? Now, there are two types of leukocyte adhesion defects. Okay? Leukocyte adhesion defects. So, dili mo pilit ang WBC sa blood vessels. And as a result, dili makagawas from the blood to the tissues ang blood vessels. And you call this leukocyte adhesion deficiency. So type 1 is associated with integrin defects. Type 2 is associated with selectin defects. So ang problem yun na associated sa adhesion is type 1, more on rolling ang type 2. Okay? Then you also have the ICAM and VCAM molecules, and these are actually associated with the integrins. Diri mo pilit ng integrins. Okay? And then, since nihunong na ang blood vessels, mugawas na siya in a process known as transmigration or diapedesis. And a molecule that is responsible for that is known as PCAM. Okay? In most forms of acute inflammation, ang first responders yun ang neutrophils. Ang first responders ang neutrophils, they usually appear in the first 24 hours. Okay? However, neutrophils typically do not last for long. So, rapid responders sila, but they usually die after 1 to 2 days. Monocytes tend to last longer. So, this is the reason why in chronic inflammatory states, ang inflammation is usually monocyte dominant sa chronic inflammation. Kasi during the first 2 days, neutrophils, mamatay siya after. Okay? So the type of emigrating WBC varies with the type of stimulus. In viral infections, lymphocytes will be the ones to go first. Okay? And usually manggod, ang lymphocytes sa chronic siya. But for viral infections, ang mag-una ang lymphocytes. In hypersensitivity and parasitic reactions, ang mag-first mo migrate would be the eosinophils. Okay? In chronic inflammation, you will see lymphocytes, plasma cells, and macrophages. This is usually after 48 hours. Now, after extravasation, the leukocytes will emigrate to the specific site of action in a process known as chemotaxis. Okay? And these molecules that facilitate chemotaxis are known as chemotaxins or chemoattractants. Okay? And we just need to memorize 3: interleukin 8, C5a, and leukotriene B4. Bacterial products may also serve as chemotactic agents. Okay? Now, essentially, I'm not going to go into the very specifics here, but essentially, the chemotaxis is brought about by conformations in the plasma membrane and the cytoskeleton of the WBCs, brought about by changes in calcium gradients. Okay, so what's going to happen is the leukocyte is going to extend protrusions into the direction that they want to go. Okay. Now, once it has encountered the infectious agent, naabot na siya, the leukocyte will try to gobble up the microorganism, and it will try to kill it by intracellular destruction. Okay? So it does so by a process known as phagocytosis. Okay? Phagocytosis involves recognition and attachment of the particle to be ingested by the leukocyte. So once ma-recognize na niya, again, changes in the cell membrane conformation, i-engulf niya, and you will result in the formation of a phagosome. And then the phagosome will bind with a lysosome to form a phagolysosome or a secondary lysosome. Okay? Now, in some instances, um, there are inflammatory mediators that can stick to the microbe, and if naasiya dito, it will make it easier for the WBC to gobble them up. These substances are known as opsonins. Okay? So if na-coat ang organism with opsonins, it's going to be easier for the WBC to gobble them up. So this is known as opsonization. And examples of opsonins include immunoglobulin G, C3, and other substances like C-reactive protein, fibrinogen, etc. So this is a picture of the immunoglobulin. Now after that, kanonia, you will form a phagosome, fuse siya to form a phagolysosome. Now, there is a disease caused by a mutation in the LYST gene, which leads to the mutations in lysosomal trafficking. Okay, the LYST gene codes for microtubule uh, assembly. Okay, now if you have a mutation of these genes, so ang mahitabo, dili ka maka-form of microtubules that will actually lead the phagosomes to the lysosomes. And as a result, makaon niya, you will have a phagosome, but ineffective ang killing kay di siya maabot sa lysosome. Okay? So this is known as Shidiak-Higashi syndrome. Shidiak-Higashi syndrome. And clinical features niya is you will be at risk for microbial infections. You will have decreased neutrophils. You will have large granules, giant granule formation in the WBCs, and the reason for that is the granules that facilitate the chemical mediators sa WBC dili man ma-break down kay guba man imong lysosomes. The lysosomes actually play a role sa maturation sa granules na nasa WBC. There is also bleeding because for the platelets to mature, you will actually break down the platelets as well from its origin cell known as megakaryocytes. There is also albinism because ang sa albinism, you will have a defect in melanin metabolism. Okay? Now, once the WBCs have gobbled the organisms up, the next step is to kill it, and there are two mechanisms for killing. One is oxygen dependent, the other is oxygen independent. The first mechanism, oxygen dependent, is the most important. This is also known as the hydrogen peroxide myeloperoxidase halide system. Okay? And it is said to be the most efficient bactericidal system in the neutrophils. Essentially, what happens here is that you will have a series of reactions wherein there is superoxidation of oxygen gas to form superoxide. You form hydrogen peroxide, and then hydrogen peroxide will react with the chloride ions to form hypochlorite. Hypochlorite is also known as bleach, and we know that bleach is a very potent antimicrobial. That is why ipahid nato sa mga surfaces, paglimpyo nato is bleach. And same thing, our body actually has a mechanism for producing bleach. Okay? You also have other substances that facilitate the killing of organisms apart from this halide system, and these include lysozymes, lactoferrin, MBPs, and defensins. Okay? You have defects in leukocyte function. Kung sa leukocyte adhesion, you have leukocyte adhesion deficiency type 1 and type 2. Phagolysosome function, you have celiac Higashi. And then defects in microbicidal activity. This is a case that I saw before. And this is actually due to defective killing of the organism. And this is known as chronic granulomatous disease. So, here, in chronic granulomatous disease, essentially, dili ka makaproduce of bleach, dili ka makaproduce of superoxides in the body. So, as a result, inefficient ang acute inflammation nimo. Okay? Inefficient ang acute inflammation nimo because you do not produce superoxides, you do not produce peroxides, you do not produce bleach. Okay? So, as a result, dili makapatay ang body using acute inflammation, and as a result, the body will try to contain it. It will shift to chronic inflammation instead. That is why you will have granuloma formation. Okay? Take-home messages here, essentially, we have talked about the basic steps sa cellular na events. So the cellular event was going to happen is because of the leakage of fluid out of the blood vessels, there is going to be margination, mukilid siya. There is going to be rolling, maghinay-hinay siya tumble across the walls of the blood vessels, and this is facilitated by selectins and sialylated Lewis X. And then finally, muhunong siya in a process known as pavementing. Okay, and this is facilitated by pavementing or adhesion, and then this is facilitated by integrins and the immunoglobulin family. Mulusot siya through a process known as transmigration or diapedesis, and this is facilitated by PCAM, and then it will go to the actual site in a process known as chemotaxis, and then upon chemotaxis, we have chemotactic agents like C5a, interleukin 8, and leukotriene B4. So once maabot siya sa site of injury, it will try to undergo phagocytosis, and it will kill the organism by secretion through oxygen-dependent and oxygen-independent mechanisms. Okay? Thank you very much for listening. See you in the next lecture.